Hi, my name is Cameron Hosking, and I'm from CSIRO's Transformational Bioinformatics team. And over the last couple of months, I've detected preliminary data that shows coronavirus undergoing active recombination within the circulating population. Recombination may occur if a host is infected with two strains of a virus simultaneously. As these two viruses are replicating in the same host cell, the polymerase complex may reassociate with the template viral RNA of the other virus. Through this process, novel recombinant genomes may be created that contain genes from each parental virus. It's important to know if this is occurring within circulating strains of coronavirus, because this affects how the virus may evolve in future, affecting decisions about use of genomic tracking and potentially even vaccine development. This isn't even particularly unlikely. Recombination is known to occur within coronaviruses, with research showing several events within the MERS outbreak and different recombination events being the likely progenitors of both the original SARS outbreak and the current pandemic. However, so far there has been little research into whether recombination is occurring among the human-specific SARS-CoV-2 strains currently circulating. So, recombination is not a new thing, and a lot of work has gone into developing methods to detect when this happens. At a high level, these recombination methods work by identifying regions of similarity between strains. So for example, they will look at strain D and see that the first half is very similar to A, and the second half is very similar to B, and hence this would be labeled as a recombination event between the three. Other events may be more complicated. For example, strain E's similarity goes ACA, and therefore contains two recombination breaks. And strain F cannot be explained by an event with existing strains, and would therefore be explained as either a recombination event with an unknown strain or occurring through normal mutations. However, SARS-CoV-2 poses a real problem to these algorithms, because while there is variation, it is comparatively small, with most circulating strains being over 99.95% homologous. So when these algorithms look at regions of a virus, they fail to find any recombination events because all regions have very high similarity. So we wanted to develop a method that could detect recombination in SARS-CoV-2 or any other virus that shows a high degree of similarity between isolates. To do this, we went back to the phylogenetic analysis and used the trees generated by Rob Lanfear to model what a normal mutation profile looked like, how often given mutations occurred, where they occurred, when they were carried forward, etc. Using this as a baseline, we were able to identify outliers. These outliers were relatively evolutionarily distant from their parents, even taking into account time duration between samples. When we found these outliers, we would then look to see if recombination could explain them better than regular mutations. We did this in a similar way as previous methods, doing pairwise comparisons with existing parents. However, instead of considering regions, we considered their sets of mutations. We tested this method on a synthetic dataset to characterize its false positive and false negative rate. We simulated the evolution of 1 million isolates with 500 recombination events. In particular, we wanted to see how our algorithm handled incomplete data, because available real-world data isn't comprehensive just due to the fact that not every isolate has been sampled. For this reason, we ran the algorithm over a subsample of 100,000 strains to simulate the dataset containing only 1 in 10 strains, to determine how this affected our recombination analysis. We detected 260 recombination events. 60 of the events were false positives, but at a very low certainty, and the other 200 reflected catching 200 out of 500 of the recombination events. The reason we only detected around 40% of the planted recombinations was because many events only had distant relatives present in the actual sampled set. However, a significant proportion were still recovered. Running the algorithm over the sequences available within GISAID's COVID dataset, our preliminary results detected 683 plausible recombination events. Several of those events are shown below. The middle sequence of each triplet is the recombinant sequence, with the top and bottom sequences being the parent sequences. The blue line follows which parent is donating which part of the sequence, but it's important to note that the actual recombination breakpoint positions are very fuzzy due to how few mutations are present. The South Korean recombination event is notable as these sequences were available from a cluster that occurred early in the pandemic, when only around 100 sequences have been sampled.